Good morning, everyone. So today we will discuss about the role of rDNA in diagnosis and vaccination. We would directly or mostly we will talk because majority of the things you already have learned before. Say, for example, vaccination, you must have learned some things. And because of COVID, this pandemic, majority of the people know many things about diagnosis and vaccination. So we will also talk in the same light. And I hope you are all vaccinated to start with for COVID, and then we will see. So the first one that I would like to discuss is, before going any further, I would want to see how rDNA is the center of the medicine in the future. So medicine would require several things. In that, we first need somewhere to have a diagnosis. Then we need to have treatment. And we also need prevention. Right? These are some of the things that we need to have. The human population in the past, say 200 years or so, has the average lifespan used to be somewhere around 34 years. And now it is around 67 or something, approximately like that. It has doubled the average lifespan of a human. And that's because mostly we have been able to prevent and treat diseases. Treatment is the points of treatment are, or the things are mostly because of antibiotics. That is one huge discovery or invention that we have made. And then we are able to treat majority of the infectious diseases, at least. Like people used to say the 5 years, right, before, when the immune system is not so active, many children used to die before they reached the age of 5. That's why the average lifespan is something like 34 years old. And they died because of infectious diseases, like for what we have vaccination for. So in general, also many people, we had a lot of diseases at different times, and antibiotics have helped us treat those diseases, and that is how we're able to lead relatively healthier and longer lives. And with prevention, we have vaccines. That is the huge impact that it had on human lives. So vaccines are a way to arouse the immune system of the host so that they would be ready for any pathogen that might be infecting, and then treat the infection or prevent the infection. And then in the recent times, diagnosis has become also the biggest tool. So in medicine, apart from many other things, at the level of molecular levels, you can think of antibiotic production or isolation of antibiotic producing genes or production of large-scale antibiotics. We have rDNA is involved. Vaccination is majority of the cases. At least in the modern vaccines, we have a lot of rDNA involved, or genetic engineering. Similarly, with vaccines, directly or indirectly, again, we have whether we want to search for the antigen or the antibody. And either of these cases, we would need large scale of those, and then we will have to depend on rDNA. So I hope you realize that rDNA shapes a lot of this in the medicine. And especially in the pandemic, imagine within 1 year, we are able to develop vaccines for a novel disease that has come by. And that is huge technology development, especially in the cases of rDNA and large-scale production, and also, of course, clinical trials and analysis. So let's look at the diagnosis first. We are not going to discuss about treatment here. These are other ways we to do. Diagnosis and prevention are what we are going to discuss, and we are trying to discuss it in light of what each of us experienced with regard to COVID. So the first thing is about diagnosis. I think we know the two prominent tests. We call it as rapid test and the RT-PCR test. In rapid test, we are testing for the antigen, whether a particular antigen is present or not. That means in rapid test, in the kit, they must be having a lot of antibodies. Where did those antibodies come from? We must have used recombinant DNA technology there as well. I hope you know how monoclonal antibodies are made. And in those monoclonal antibodies, so antibodies should have been produced in large scale, right? So in that, hybridoma technology will be used. A antibody producing cell and a cancerous cell, they will be fused to make a hybrid cell, which has the capacity to produce the antibody and also continuously grow. And I hope you remember HAT medium. If not, you might want to look at it. It's interesting as well. So sometimes we may have to clone the antibody and then try to produce the antibody in large scale. That is another strategy. One can also do it. But this is a much better and easier one, or the traditionally done one. So in rapid test, we are finding if an antigen is present. What is an antigen? Antigen is say, if there is a coronavirus, all the particles that they are present with the RNA genome and several other components, spike protein and so on. All the factors that are present in the virus that are not part or not similar to that of the human proteins or any components, right? So basically, we will just look at it as this is a, say, human body. And immune system, like we discussed in CRISPR-Cas, should be able to differentiate what is self and distinguish what is non-self from that of self, or what is foreign and what is self. So the immune system also has a similar strategy where during the maturation or so of the antibody producing cells, if there is any cell that is B cell that is producing antibodies that are targeting the self antigens or say proteins or other components, those cells will undergo apoptosis. So the human body, so the immune system is working by random generation of diversity, right? It is random generation of diversity, and all those that are self are eliminated. Any antibody producing cell that is against the self components will be eliminated. So what is left is antibodies that are specific to any protein that is but not of the self components. So all those, if a virus has entered into our body, that is not self, right? That is foreign, and all those can be recognized as or called as antigen. We will say antigen when we have an antibody produced against it. If there is an antibody and it can recognize a protein component, then we will say the foreign component, we'll say that as the antigen, and the other one as, of course, the antibody. So when an infection occurs, there are two things. One is in the body we have antigen, that is the virus here, and if the host has produced antibodies against this antigen, then we will have antibodies against that particular antigen. So how do we diagnose is to check for antigen is one strategy. To check for those specific antibodies produced against that antigen is another strategy. The third is doing the molecular tests, right? That is, if it is a infectious agent, something like COVID or any bacterium, we can test if that any genetic information pertaining to that particular, that are specific to this, the viral or bacterial pathogen, are present or not. If they are present, that means the pathogen is in the body. That is how another way of diagnosing it. So broadly, there are three categories, right? Whether you want to find out, you want to identify the antigen, I mean, find if there is an antigen or not. You want to find if there is the antibody generated against the antigen. And the third is trying these molecular tests, trying to see if a particular gene is present or not, DNA, genetic information is present or not. And so there are, these are the tests. I hope you know about the first one is the antigen test, rapid test, where they have a strip. I guess many of you have undergone through that particular test, where they take a swab and insert into your nose and collect. That is where the likelihood of the pathogen is there in higher concentration. So the swab is taken, and that swab is using a slide-like thing. They would have allowed, there is processing of it a little bit, and then not so much. And then we have whether the strip has the antibody against the spike proteins, the S protein, right? That is on the outermost surface. So the strip has antibody. We are collecting the virus, and the spike protein, if it is present, would have interacted with the antibody that is present in the strip, and it would give an indication, usually a color-based ones. And if it is positive, if it gives a color, then it is positive. If it doesn't give a color, then it is negative. There are problems associated with this. If the processing is not accurate, processing of the sample is not done well, or if the amount of antigen, the virus, is not high in concentration at this location at the time of taking, this person may be infected, but to have a full-blown symptoms and accumulate in the nose, nasal cavities, and nasopharyngeal places, it might not have been in appropriate concentrations. That is also another point that might affect the result of it. The third is we are having antibody-based test. That means this antibody is based on a protein, so its storage and shelf life should be appropriate. Okay, so the first thing we'll also say is collection. We have to also take into account the collection. If anybody has gone through the testing to put them and to collect from your nose, I went through once, and it's horrible. And people usually keep moving, and it is actually a tedious process. If the sample is not collected in appropriate amounts, then there is a problem. The processing, if it is not done well, whichever the storage of the sample and then the processing until it is added onto the strip, if it is not done well, that is another problem. Within the sample, assume the sample is taken enough. Within that, if the COVID virus, for example, in this case, is not in appropriate amounts, that might also affect. And storage of the kits is also an important thing. So why I'm spending this much time is rapid tests are troublesome in many cases. There are advantages of using rapid tests. It's easy, it's fast, it is inexpensive, And it can be useful when we are doing with many, if we have to do lakhs on thousands or crores of people, then rapid test is the way to go, because something like our country immediately cannot afford all of it. So if a positive test is given, then it is confirmed that that person is positive. If a negative result has come in the rapid kit test, we are not very sure, because there are too many factors that can interfere in the negative result, right? So negative doesn't mean this person is not infected. Okay, maybe we can then probably shift to probability based. Maybe these are 70%, you can say 50% negative and 50% we don't know. All right, negative means we don't know. So I have my own cousin who has had a friend with positive case, and he got a friend got positive, so he was admitted in the hospital and taken care of. My cousin got negative, so he was at home. Because he got negative, he was thinking it's in a village, and he was thinking it's negative, so whatever cold and other things, he was thinking it's typhoid, other thing. He had been at home for too long until it became very serious, and then joined the hospital. And unfortunately, he didn't survive. He died young. um, why I'm bringing this up is about our clarity of thinking, right? And what we trust, what we don't trust is also a problem. I mean, that is where everything comes by. The person who got positive is saved. That person got medical attention early age because he was positive. He was taken to hospital, and he was saved. The person who got negative eventually lost life. It is about my cousin's trust on this, or probably misinformation. They didn't have much of a knowledge, and it's in the village, rural setting. So that is one of the risks that are associated with these kinds of things. So we have to always think clearly and act appropriately. So the point, other ways of doing it was famous. The other one is trying to see for antigen, whether antigen is produced against the COVID viruses. So in that case, we must be having, if it is done by Elisa or so, in that case, the strip would be having the antigen, something like the S protein. A large quantities of those S protein must have been cloned, produced in large quantities, and the wells should have been cloned with that. So I hope you remember the Elisa methods. The concept is here. If we take out the blood, take the serum, and then try to see if there is a positive reaction between the antigen and antibody, then we say it is positive or it's negative. In many of here in this case, we are taking the blood. When we are testing the antibodies, we would have taken the blood, right? So usually, if there is this is much more reliable, but then we have to take blood, and we have to process this sample to get rid of all the other stuff, interfering stuff, and try to get serum that is ready to interact with the antigen in the kit. Okay, and then we have the RT-PCR method, the molecular test method. That is very reliable, but very expensive, very slow, and it requires a lot of technical expertise, training, and so on. I think we remember that it is by real-time PCR or qPCR, you can say. The sample is collected from the individual, and the sample is processed. And the process, in that case, we would have isolated the RNA, and then we would have used primers that are specific to the, we would have performed reverse transcriptase by PCR, followed by the normal PCR. I mean, PCR along with real-time reading. So the primers should have been specific to COVID. So those primers should not have any other interferences in our or non-specific band should not come. So people have identified or sequence based on the sequences, they've identified some of the primers that are highly specific to COVID, and then they were used. This one is much more reliable because it is, even if there are few copies of RNA, we can detect them. Okay, so that increases the reliability of the method itself. So it's because there is amplification PCR based, and we can do real time, and there are several ways. I hope you remember how to see the real time PCR, the CT values and other things. So if there is a product and it is coming early, then we can detect whether we can detect the amount of COVID within the sample. There are, of course, other ways of doing which don't require the molecular techniques, neither genetic information or the proteins. It is more based on the chest X-rays and CT scan, right? That is another way of doing it. So all the things in I wanted to mention this is we have to always keep in mind what is correct and what is wrong, our ability to think. If we are developing a diagnostic kit in tomorrow's world, we should then keep in mind several concepts, right? The points that we have to remember, if we have to do, is it should be simple, but it should be reliable. It should be able to, we should be able to produce it in mass production should be possible. Storage, maintenance, and operation should be easy and should be portable. And there could be other ones. The first objective is reliability. Okay, our priority is reliable. That is the purpose. We want to know because it could cause some serious injury, or it's about life and death. So reliability is the first priority. But again, if there is something that is 100% reliable, say RT-PCR, 100% reliable, and say rapid test is, say, maybe 70% reliable or 60%, I'll just put it down. Then let's see what is gaining more marks. Which one is simple, RT-PCR or rapid? Rapid is simple. In reliability quotient, you can say RT-PCR is better. It should be inexpensive. Storage, mass production, and this is all right, we can just say. Storage and operation, no, this is easy. Portable, not so much. You have to collect samples and produce and send it to a center that is actually performing the RT-PCR, whereas rapid tests can be done then and there. Which one is inexpensive? This is expensive. This is inexpensive. So this also gives you, in total, if you look at which one should we go for, is if our population is less, then this is the best thing, because population less and we are rich. Okay, that is also one thing we have to count. Then we can go for RT-PCR because many things can happen much faster and there are not so much of burden to be tested, right, in terms of population. But if the population is very high, and or if there are very poor financial situations, then the rapid test is better. Okay, at the end of the day, we want to contain or somehow be able to manage the pandemic situations. If it is a 1-2 cases in non-pandemic situations, that is all right. Any of these should be fine. But if you want to do, if you are doing, how can we increase the reliability of this rapid test? We have to increase the n, that is the sample size. What I mean is, maybe we should try to take two or three rapid tests and see whether we are getting we are getting negative or not. That could probably increase the reliability. If you are doing the same test to the from different brands, similar principle on the same person and getting same results, you can increase the reliability by increasing the sample size. These are other ways of how we can improve this. Okay, I hope this scenario is clear, right? Good. The next one is vaccines. In vaccines, I hope we have read in immunology about the importance of vaccines. There are many things. We have the traditional one is killed whole organism. That means somehow we have to put in the antigen into the body, so that in the body we will be able to select for antibodies producing cells that have specificity towards the particular antigen. So the one that is already being put there should not be any danger. Okay, it should not be harmful to the person. The only purpose of why we give this is to trigger the immune system against it, so that if we happen to contract the disease, we will have effective treatment against it. So the killed whole organisms is the traditional one. You culture a large batch of the pathogen, you heat kill it or chemically kill it, then purify or probably treat it further so that there will not be any kind of problems, and then we put them into individuals in small doses, so that the antibodies will be brought. There is one problem. In one of the cases, I can't remember which country it is, there was a missionary who have administered polio vaccine to certain individuals, and many of the children there got polio. They gave vaccine, and the individuals, instead of being protected, they were infected with polio. So the way we use these is also a little bit of troublesome. We need to keep. There are a lot of issues that happened in the previous times. So apparently, I can't remember. This was a long time story. I don't know if there are any further developments. So they were heat killed, and then they were transported in trucks, and probably, I mean, this is just a theory, there was movement in the vials, and then somehow a few of the viruses got rejuvenated or formed a particle that is infective, and that may have caused the polio in those children who are apparently or supposedly vaccinated. So there is always a risk with this issue, and one has to be very sure about it. The other one is live attenuated, where say for example, we have a virus or that is entering into the cells using a receptor. Then we make a similar virus without that particular receptor or a protein that is actually causing it to be virulent. So we're making, we can replace those genes and then produce non-virulent type, so that the antibodies will be raised against that attenuated one, and then or live ones or non-virulent ones, so that we will be protected from the virulent one as well. There are also other ways of live recombinant, where we can take a virus that is actually not so harmful, and put in one of the proteins, genes from the pathogen. And this is when this is expressed, the antibody, it will be expressed, and you can use this as an as a trigger to the body to allow it to be produce antibodies. There are also DNA vaccines where you directly take a DNA and put in the gene of interest or the pathogen gene, virulent gene, and give it to the individual. Some of the cells may take up this DNA and they will transcribe and then translate the protein and may secrete it out into the blood. So the immune system will develop antibodies against that um, particular antigen that has been put. There are risks with this because we should know how, when, and where it is integrating. And if it integrates into a wrong place, there is a probability that it might cause cancers or some other genetic problems. So this is a quick review of many things about vaccine that we discussed. Now we will look at what is with the vaccines. And just before going there, I also want to discuss about the once we produce a vaccine, say for example, a vaccine has been designed and it was offered, we were all afraid to take the vaccine, either because somebody says this is not good, or there are a lot of ill effects, and there are many things we want to do. If we want to do anything, we feel a little bit of anxiety, and we feel anxiety, and if somebody else also shares their anxiety, and then we become too sensitive about it, and that's understandable, and it's very common and natural. And so what we need is, first thing, as I was talking before, the vaccination is little touchy, in the sense that even in the US, there are people who think that vaccination causes autism. Let's say somebody opines, and especially somebody with a lab coat, or so scientist like thing, or influential or popular person says the thing, people who do not have any idea or who don't belong to that field may become highly anxious and then scared to go ahead with it. That's a normal response. There is nothing intelligent or unintelligent about it. It's just a normal response. So there should be many clinical trials that should go into before an approval for a vaccine is being made. Preclinical stages. And nowadays, people have become very smart that they are looking into the results of this. Okay. So phase 1, we will try about dozens of volunteers in a period of 6 months. Phase 2, about hundreds of volunteers, and phase 3 is thousands of volunteers. And when the vaccination goes through all these phases, if they pass here, this is allowed. Incrementally, they will allow, and only then we have approval of it. And we did hear many stories, right? Somebody got vaccination, and in 4 or 5 days or so, the person died. So the person died because of, say, heart disease. So vaccination, what would we believe is the question again. This is again a case of clarity of thinking. I mean, I'm not saying anything is right and wrong. I'm just trying to see that we all be able to think in proper ways. So what is the cause of the death of this person? The way the news will be told is, he has been vaccinated 3 days ago and he died of heart disease, right? All of them are facts. Vaccination, he has undergone to, fact. He died, that is a fact. And he died of heart disease, that is also fact from the autopsy. So the problem is it is easy to the many people who hear that news and who don't have that clarity of thinking will associate vaccination with COVID thing has caused heart disease and the person died and causes death. Well, there is still, I'm not saying it's not possible. I'm only saying how to think. Did we also consider other factors, if there are any? Did we see if this person has any comorbidities or other things like BP or any lifestyle diseases, however, his diabetes and many other factors can also cause heart disease and then eventually death. Did we eliminate all the other stuff? We did not. But the news would only say these three factors. And we are at the recipients and we are already anxious. And a news like this will cause us more anxiety. So I wouldn't know what has caused the death. Is it vaccine, or if the person already has any other issues with that are not known to us at this time, right? So we need to keep that in our mind. And again, it's an issue of clarity of thinking. Our clarity of thinking is mostly dependent on the information available. Information that is available and our competence to look at information, right? Our clarity of thinking will depend on these two, at least our competence to look at information and also inferences and to draw inferences and with certain degree of reliability. Okay, with COVID, there are different types of vaccines that people have developed, and some of them are inactivated, attenuated viruses. They culture the virus, inactivate them, and give it. Covaxin is one of such example. Viral vector, non-replicating. You can take a viral vector into which they genetically engineer and put in a spike protein gene for spike protein, for example, which is relatively harmless and which will produce coronavirus proteins and we will get immune response to it. I think Covishield is based on that. There are also other types of where you can just produce large quantities of, say, for example, S protein or spike protein and give it into the individuals. As I described a little while ago, giving DNA with a gene, with a gene for COVID-19 antigen. There are also a new version in a way is the RNA-based version, because as I said before, giving DNA vaccines could cause problems. In the sense, if we give and the DNA is integrating with any part of our genome, and if there is a possibility of where it might cause or activate oncogenes, that is a serious risk we have. So instead, we could try mRNA-based or RNA-based vaccines. And this is a more likelihood. I will also use this space, say, for example, this is the nucleus, and transcription happens in the nucleus. So if we were using DNA vaccine should go into the cytoplasm and then go into the nucleoplasm to be transcribed and then translated. Whereas if we are using mRNA vaccine, say, mRNA vaccine will go, translation occurs in the cytoplasm, so it can produce the antibodies, the antigen, which could be secreted out. So in comparison to DNA vaccines and RNA vaccines are much safer, theoretically at least, because they're RNA, they're less stable, the probability of integration is very less, and they don't have to get transcription and all those things. But there is also a problem, as I said, which is RNA is not very stable, which means they will be degraded much faster. So we need to give more. Giving the protein subunit or giving the genetic information, as in this case DNA or RNA. If you give 100 proteins, that is what is present in the body. If I give 10 mRNA, each of them is translated 10 times, you will get 100 proteins, right? So the dosage could be reduced if you are giving the information, because they can be either transcribed and translated or directly translated. So there is a probability for larger production of those proteins. So those are some of the advantages. If we are proving any of these things, okay, even the other ones as well, apart from the inactivated or attenuated vaccines, if we are using any of them, we will have to construct a recombinant DNA and then be able to produce, ensure that the antibody is produced in sufficient quantities in each of these cases. So there are many other vaccines, are a little bit of history. You might have already learned it, and which I'm not going to discuss, but it's a good thing to have a look at. I'll stop here. If you have any questions, I would like to discuss, and I would also like that you share some of your experiences with regard to COVID, and I will try to share what I know. Anybody? If you don't have any questions, yes, Parvati, go ahead, please.
This is not typically an experience, but as you
Okay.
said, anxiety, like there is this Omicron strain which is getting reported. So we would love to hear from you. What is your take on it? What are your thoughts about it, sir?
I don't have any. I mean, I don't have any more anxiety about it. If it is there, we will have to go back into our house. That's it. So, what we should think of it is also this way. If it is very dangerous, highly virulent, that means if it infects a person, it kills the person immediately. And there is another thing, the ability or speed with which it spreads. What do you think virulence and spreading are? If there is high virulence, if it is killing somebody very fast, then the rate at which it will spread will be lesser. Okay, if it is highly virulent, killing somebody immediately. But with COVID, we have different problems. We have high mobility. We are densely populated, and mobility is high. And these are the problems. And of course, there will be hygienic issues. And hygiene is an abnormality, isn't it? What I meant is, it's not inherently built into our system. We would have been like animals without brushing or sneezing openly. We would have done many things as such. So that is, we haven't learned, not everybody has learned the hygiene as much as we should have done. So these are the problems we have. So right now, and we are so much bothered or affected by COVID because it is airborne, and people will sneeze, and we are living densely, and we are moving too much, too far, and too often. So the rate at which we are spreading COVID is very high. Okay, that is a typical pandemic situation that happens. even if it is Omicron or so, I think somebody was telling me that it's not as dangerous as this. It has, I didn't go through the genetic nature, or I just looked at it. We are all, majority of us are vaccinated, although there are several mutations in the new strain that is coming up. We also have several, say, when there is an antigen, we will have several parts, and each of them is an epitope, right? Antibodies could be generated to this, and so on. And this also calls for, in the same case, Covaxin and Covishield. Covaxin is for whole virus. Covishield, I think, is for the S spike protein, I think. Right? So antigens produced against spike protein, they are large in number, and it is very effective, Covishield. Covaxin has the breadth, in the sense it's not only for spike protein, it could be for many other surface proteins that are present in Covaxin, of COVID virus. But apparently, I don't know the details. We all have to look at the literature properly before saying. That's why I don't want to speak about it, but we'll just share just as a thing. So if we have an antigen, and apparently Omicron has several mutations in many of these epitopes, but I'm hoping that there are some epitopes still that could be targeted by the vaccines that we already have taken. So the rate at which it will spread may not be as much, but this is, of course, the problem, and this is the second problem. People are going, and we already stopped using the masks and other things. It has become difficult also to use after being 2 years with the mask. But I don't see there will be big issue as far. I think it will be contained because we are vaccinated, and I'm hoping, okay, I'm not sure. I'm hoping that there are still some of the areas where the mutations have not have been done, and our immunity that we have is still effective. Let's see. Maybe we'll all be given a booster dose of vaccine if required at some other point. Yes, we don't have to worry. But the problem is the society, the general public. They don't. The public doesn't care as much. Already, people have stopped using masks, and many things are happening. Where in gatherings and unnecessary travel, we want to have a life how it was before the pandemic, but I guess we should still be careful about it. Yes. What do you have any idea, Parvati? Did you read anything different, or what is your opinion?
So as you said, there is no much of a data to make a
Yeah,
credible.
that's
Yeah.
right. Yeah,
But
but
as you
otherwise too, we
said,
have to
people.
yes.
Sorry, sir. Like as you
Yeah,
said, there is no much awareness right now. Like people are so restless to get outside, and they don't
yes,
even care to wear masks. So that is really
yeah.
concerning.
Awareness is the wrong word to use, right? They know it. Everybody knows it. We don't care much. The second word that you also use, that word that they don't care, that is a correct expression. Yes.
Sadly, yes, sir.
But I mean, it's also understandable. But see, I would also want to bring you one thing. We have 5 minutes. So if we want to cultivate any habits, okay, we and then we maybe a teenage, and then an adult, and we will remain like that. Maybe we'll grow fatter, and that's it. The rate at which, at the best time for creating habits is at this level. And if you have any children at your home, please teach them how to sneeze and cough. how to use their elbow or sleeve to cover up their sneeze and coughing. Every class that I go, I usually try to impose that in as a kind of thing. And if you have any children at your home, brothers, sisters, some among your family members, please tell them to do it and try to create that habit in them. As such, right away, there is no hope for, I don't see any hope that any of the elders, the grown-ups, will ever learn that. It's impossible. It's almost impossible. You can see people who will, until then masked, they remove the mask, sneeze out, and put up the mask back, isn't it? So it is our, as biologists, it's our responsibility and to live the science that we are learning. And the best way to create good habits for the social uh, hygiene is to, the target group is the children. Please try to do them, make them sneeze and cough twice, thrice. You tell them, correct them, and you have created a habit, the habit in them. Then you have made a good service to the human society or species. These are the problem. This is the problem. Even at my home, in my native place, my mother and all, it's very difficult to bring in those habits because it's. I don't know how many of you have got used to it and how many of you are following it. It's. I think we also have a responsibility to um, educate the society, and this is a dangerous group to educate, because if you go and say, they will get upset, they will get angry with you sometimes, and they will actually deny, or it will be very involved with a lot of friction. Whereas children, we have an advantage, and we can say it softly and funnily. We can make them do it. We can playfully suggest that they should do it and make them imitate these actions of how to cover up if they don't have a mask and the sneeze is coming up or a cough. How to contain it immediately? Just use use the elbow or the sleeve so that we don't spread that out. Not even the hands. So if we can take that and try to do this is our target group. Maybe in 10-15 years, the whole society will be filled with people who will be following these very well. Okay.
Yes, sir.
Yeah. Okay, any other questions? And tomorrow we have a class, and apparently, at least for this session, that will be the last class, isn't it? I think I have covered almost all the syllabus. Just NGS is present. NGS is left. I will take it up tomorrow. Just the basics we will discuss. And I would want you to watch the videos that I already shared with you. Watch NGS on that. They are actually in much more detail. I will not go into that much, but we will just discuss one or two principles of how it is done. The practical lab one. I'm not going into the analysis, which is a lot of bioinformatics work that we will not be able to discuss as much. It comes under bioinformatics. Okay, but we'll see the general principle.
Sure, sir.
Yes. Thank you, everyone. If you have any questions, you can let me know. And I think we will have one class per week from the next week onwards. We will discuss more problems during that time. Okay. Take care and have lots of fun.